Assalamu alaikum. Today we will discuss the second part in this chapter, which is cell as the basic unit. We will define a cell in this part. We will discuss the types of a cell, which are prokaryotic cell and eukaryotic cell. And we will discuss examples of prokaryotic cell, which is bacteria, and examples of eukaryotic cells, which are plant cells and animal cells. So let's see how do we describe a cell first. So let's see, a cell can be described as a basic unit of structure and function of all living things. Okay, so all living things are made up of cells except viruses. Viruses are only non-cellular. Otherwise, bacteria, plant cells, animal cells, fungi, algae, or any other kind of life, they are all made up of cells. There are two basic types of cells. Prokaryotic cell and eukaryotic cell. Prokaryotic cells are simple in structure than eukaryotic cells, and they possess some characteristic features that may be used to identify prokaryotic cells from eukaryotic cells. We will discuss a comparison in between prokaryotic cell and eukaryotic cells, but let's see the most prominent difference in the prokaryotic cell and eukaryotic cell is that prokaryotic cells lack nucleus and eukaryotic cells contain nucleus in them. We will discuss the other features that differ in both prokaryotic and eukaryotic cells. But before that, let's see an example of a prokaryotic cell, which is a bacterium. So we'll discuss the structure of a bacterial cell first. So here we have a bacterial cell drawn in this drawing. You can see there are some structures that are always present in bacteria or in prokaryotic cells, but there are some structures that may be present in some bacteria or may not be present in other bacteria. Features that are always present in bacteria include this outermost cell wall. This is, cell wall. We can, so this is cell wall. We have represented the thickness of the cell wall with two lines. Okay. And towards the inner side, you can see there is another line. There is another line, which is cell membrane. And you have a circular DNA molecule in it, which is a naked circular DNA. And you have cytoplasm here. So cytoplasm here. Okay. And there are 70S ribosomes here. So these are the structural features that are always present. So if we label them one by one, however, this flagellum is not always present in bacteria. This may be present in some bacteria and may not be present in other bacteria. The next is, you see this cytoplasm. Cytoplasm is found in all bacteria. Then we have cell membrane, which is also present in all bacteria. We have this slime capsule, which is also not found in all bacteria, may or may not be present in some bacteria. And then we have, DNA. It is circular DNA, and it is naked DNA, which is a characteristic feature of uh, prokaryotic cells or bacteria. And then we have ribosomes. These are 70S ribosomes. We will discuss what does 70S ribosomes mean. And then we have this cell wall. So if I, draw this cell wall with this green color, so this is a cell wall which is right outside the cell membrane. And then further on outer side, you can see this is, this part is the slime capsule. Slime capsule is not found in all bacteria. So it is not a characteristic feature of all bacteria, right? So I have colored it differently. So these purple structures are not always present in bacteria. There is another feature which is known as plasmids. So these plasmids are also found in some bacteria and are not found in other bacteria. And then you can see the pili. This is another structure which is found in some bacteria, and they help stick these bacteria to other surfaces. Then diameter of bacteria ranges from 3 micrometer to 5 micrometer, and they divide by binary fission. There are two processes of cell division. One is known as binary fission and other is known as mitosis. Mitosis is a type of cell division in eukaryotic cells, whereas the binary fission is a type of cell division in prokaryotic cells or in bacteria. You can have a look at the prokaryotic cell. There are no mitochondria present. There are no, the double membrane bound other cell structures. There are no chloroplasts present in it. No Golgi bodies, and no nucleus or nuclear membranes present in these prokaryotic cells. Okay, so let's see. Then we have the next slide is eukaryotic cells. So eukaryotic cells have a nucleus present in them. As I said earlier, nucleus is present, and you can divide eukaryotic cells into many categories. One are animal cells, which have a diameter of around 20 micrometer. And then we have plant cells, which are larger than animal cells, and they have a diameter of around 40 micrometer. Okay. They divide by binary fission. Sorry, they divide by mitosis. Okay. During growth or an asexual reproduction, whereas they divide by meiosis during the gamete formation. At this point, we will not discuss, the meiosis. Meiosis is an A2 syllabus, so we'll discuss in an A2. Then there are two examples in our syllabus. We have animal cells and plant cells. So let's see the components of cells first, and then we'll have a look at these animal cells and plant cells seen under a light microscope, scanning electron microscope, and a transmission electron microscope. Components of a cell, a cell consists of cytoplasm plus cell membranes and plus cell wall in case of plant cells. Cytoplasm, on the other hand, is made up of a liquid ground substance which is known as cytosol. So this is a solution or a liquid part of a cytoplasm in which the living components are embedded. These living components are known as cell organelles. And there are non-living components also found in cells which are known as cell inclusions. Cell inclusions include like granules of maybe starch, glycogen granules, fat droplets, or some crystals of the salts, whereas the cell organelles are the living components, and we can divide them into three categories. One are the single membrane bound cell organelles, the other are double membrane bound cell organelles, and the third are without membrane cell organelles. We'll have a look at them one by one, starting with the double membrane bound cell organelles. Double membrane bound cell organelles, double membrane bound cell organelles include nucleus, chloroplast, and mitochondria. Okay, so let's see the structure and function of a nucleus first. So a nucleus, as we discussed earlier, is found in eukaryotic cells only. It is not found in prokaryotic cells, and the size of the nucleus that varies depending upon the function of the animals and plant cells. However, on average, a nucleus has a diameter of more than 10 micrometer, and it is a double membrane bound cell organelle. It has an outer membrane. You can draw this, an outer membrane. This is an outer nuclear membrane. Then we have an inner nuclear membrane. Okay, so this is inner nuclear membrane, and collectively these two membranes are known as nuclear envelope. These are both partially permeable membranes, so they allow certain substances to pass through them, and they do not allow the other larger molecules or particles to pass through these membranes. Okay, so let's have a look at the differences in between the inner membrane and outer membrane. The outer membrane has small granule-like structures present on its surface. These are known as ribosomes. These are ribosomes. Whereas the inner nuclear membrane doesn't have ribosomes present on it. This is a smooth membrane, whereas the outer membrane has ribosomes attached on it. On the other hand, the outer membrane is continuous through an extended network of channels, which is known as an endoplasmic reticulum, or which is known as a rough endoplasmic reticulum. So this rough endoplasmic reticulum also has the ribosomes attached on its surface. So that is why this is known as RER, or rough endoplasmic reticulum. Rough endoplasmic reticulum. So inner membrane differs from an outer membrane in a way that inner membrane is smooth, doesn't have any ribosomes attached on its surface, and then also it is not communicating with the endoplasmic reticulum or rough endoplasmic reticulum. Then between these two membranes, there are pores present. These pores are called nuclear pores. So they allow movement of substances into and out of a nucleus. There are certain substances that can move into and some substances that move out. So nuclear pores are for the movement of materials into and out of a nucleus. So this is a nuclear pore. We also have a spherical densely stained body, one or more densely stained bodies in the nucleus. These are known as nucleoli. Singular would be nucleolus. And this nucleolus is involved in formation of ribosomes. We also have DNA covered with histone proteins, which is loosely coiled and is present in the nucleus. When a nucleus is not dividing, these are thin threads visible under a light microscope and is known as chromatin. As this cell begins to divide, this chromatin coils and condenses and becomes visible as thick threads called chromosomes. Nucleus is involved in protein synthesis, or it controls activities of a cell by controlling the synthesis of different proteins. So it contains genes on DNA which control polypeptide synthesis. Okay, how do they control polypeptide synthesis? We'll discuss in nucleic acid and transport, nucleic acid and protein synthesis. Let's see then mitochondria. Mitochondria are found in both plant and animal cells. They are found in all eukaryotic cells, and they are also known as powerhouse of a cell. They are involved in formation of an ATP during aerobic respiration. So mitochondria have a role in aerobic respiration in formation of ATP. Which is provided for various activities of a cell. If we look at the structure, they are sausage-shaped or rod-shaped. They are also double membrane-bound cell organelles. The outer membrane, in this case, is smooth. This is an outer membrane. Then we have an inner membrane. Inner membrane is thrown into finger-like foldings on inwards, and these finger-like inward foldings are known as crista. Singular is crista and plural is cristae. So this is an inner mitochondrial membrane. There is a space present in between them, which is known as an intermembrane space. The inside of this mitochondrion is filled up with a fluid-like material, which is known as mitochondrial matrix. This mitochondrial matrix contains circular DNA, it contains 70S ribosomes, and it also contains enzymes for stages of aerobic respiration. So aerobic respiration takes place on inside mitochondria. ATP is made during aerobic respiration, which may be used by the cells for an active transport, for movement of the vesicles, or for other activities in the cells. So we'll discuss the role of ATP in some other lesson. Now let's see the structure of a chloroplast. Chloroplast is found in the plant cells only. It is found in mesophyll cells, and it is also found in guard cells. If you look at a leaf, cross section of a leaf, many leaf tissues don't have any chloroplast present in them, like epidermal cells don't have any chloroplast. Vascular bundle doesn't have any chloroplast in them. It is only the mesophyll cells and the guard cells. Okay, these chloroplasts have different shapes in the different species of plants. The shape of chloroplast is sometimes used to identify the species of a plant. However, they may be disc shape or they may be an oval shape structures which have a diameter of around 4 micrometer. So let's see. This is an outer membrane. And the inner membrane is, this is an inner membrane. Inner membrane. This inner membrane is folded, and these folds are stacked one above other, making a stack of this folded membrane known as granum. This is singular, and plural will be grana. And there are many grana that are present in a chloroplast, and these grana are connected together through membranes. These are known as intergranal lamella. Intergranal lamella. The folded membranes in the grana are, as I said, stacked one above the other. So if we look at this stack of folded membrane, one of this fold of single membrane is known as thylakoid. So you can see this single folded membrane, single fold of membrane is thylakoid, and stack of many, this is stack of many thylakoids, which is known as granum. There are light absorbing pigments present on this thylakoid, and they absorb light energy, and ATP formation and light dependent phase of photosynthesis takes place here on thylakoid or on grana, whereas the other light independent stages of photosynthesis that take place in the fluid inside the chloroplast on outer side of the grana. This fluid is known as stroma, and the stroma has circular DNA, and it also has 70 S ribosomes. So chloroplasts are site for synthesis of ATP by photosynthesis. we have a whole chapter on photosynthesis in Na2, and we will discuss photosynthesis in detail in Na2. And this is just enough for AS students. So that's all about double membrane bound cell organelles. In the next lesson, we will discuss the single membrane bound cell organelles one by one.